My name's Annika Lucassen, and I'm really honoured to be chairing this session this afternoon after some great sessions this morning and a lovely lunch. So this is introducing the fourth paper of the meeting, and I'm delighted that May Rizig is going to be speaking to us. Now, May studied medicine at the University of Khartoum in Sudan and completed her general medical training in several London hospitals. She obtained a master's degree and a PhD in molecular genetics from University College London and a diploma in clinical neurology from the UCL Institute of Neurology and is a member of the Royal College of Physicians. Currently, she's Senior Clinical Research Fellow at the Institute of Neurology and a Specialist Registrar in Neurology at Imperial College London. Her research interests are studying the clinical and genetic diversity in Parkinson's disease and other neurodegenerative conditions in Black populations in sub-Saharan Africa and the African diaspora in the UK. She completed a 3-year postdoctoral clinical research fellowship with Professor Nick Wood, the UCL Institute of Neurology, recruiting and characterising a large cohort of patients with the rare LRRK2 gene mutation. She's led the development of an international collaboration of neurologists and neuroscientists from 12 African countries that now has a membership of over 110 clinicians and scientists. They're collecting DNA from Parkinson's disease and match controls throughout Africa and have developed links with other national and international Parkinson's disease programs. So May, that's a fantastic CV to be giving us a paper on ethnic variabilities in neurodegeneration. What do we know about the genetics of Parkinson's disease in African populations in comparison to other ancestry groups? May, the floor is yours. Thank you very much.
Okay, so thank you very much, Anna, for the introduction, and thank you very much, everyone, for inviting me to be with you here today. So my name is May Rizig. As Anna introduced me, I'm a neurologist, but I have huge interest in neurodegeneration, specifically in underrepresented groups. I work with colleagues across the African continent to look at the genetics of Parkinson's disease in Africans. So the title of my talk today is Ethnic Variabilities in Neurodegeneration. What do we know about the genetics of Parkinson's disease in African population in comparison to other ancestry groups? So the reason why I've been invited today is because the last 7 years of my life, I devoted my weekends, my holidays, most of my time to develop this consortium. So the consortium is the first Pan-African consortium dedicated to studying Parkinson's disease in African populations. And it has several components which have been mentioned this morning during the talks from Ambroise and others who they work in Africa before me to address the diversity in other conditions. So we incorporated clinical and genetic research with other components such as education and training, capacity building, and public involvement. We're still very, very young consortium. We started in 2019, so it's about 4 or 5 years of work, but hopefully I will convince you that if you really dedicate the time and the effort and you have the right links and collaborations, even in a very short space of time, you can achieve a significant advance. So through my talk, I will try to answer certain questions. So some of them I've asked myself on the beginning because they were quite confusing to me when I started doing this type of research. So I put them, I listed them, and then I would just read through them to reiterate some of the facts even for myself and the audience. So I will start by defining what I mean by ethnic and ancestry groups and what do we mean by Black population, where do they live, and what is the number of people who can identify themselves as Black from people from a Black ancestry groups, and what is the global burden of neurodegeneration, what is exactly Parkinson's disease, and what do we understand about the genetic factors which are contributing to Parkinson's disease, and what are the disparities in Parkinson's disease. especially in underrepresented groups and in Africans in particular. And then I will end my talk by debating the importance to consider ethnic variability when studying neurodegeneration. And I will focus on Parkinson's disease for that purpose. So the definition of ethnicity and ancestry, it was very, very confusing. I've heard a lot of people debating about it. I generally accepted the definition which I wrote in this slide, and I will read it as I wrote it. So ethnicity is defined as the social group a person belongs to and either identify with or is identified with by others as a result of a mix of cultural and other factors, including language, diet, religion, ancestry, and physical feature, traditionally associated with race. So if we start to think about this definition, to me, it means more of a social construct rather than a biological construct. And when you looked at the definition of the ancestry, the definition are the people that an individual is biologically descended from, including their genetic relationships. These genetic information can be combined with historical information to infer where an individual's distant ancestors lived. So to me, it's more of a biological construct rather than a social construct. Whatever that we use as a definition for ancestry or ethnicity through my talk, I will use these two terms interchangeably to speak about the genetic origin of a person or a population. So taking that definition into account, if we speak about populations from African ethnicities or ancestries, we should consider them as a very diverse group of individuals with various genetic admixture, ethno-linguistic backgrounds, and cultural origin. Where do they live? They live everywhere. So when we talk about Africans or population from a black African ancestry, we think about Africa only. This is not true. By default, the majority of them live within Africa, so which is the home for about 18% of the world population. This is about 1.5 billion people. This percentage is expected to increase to 26% by the year 2050. And Africa itself is a very complex, not just from the genetic background, even from a geopolitical background, is composed of 5 zones, 57 countries. And outside the continent, 250 million people who they identify themselves as people from an African ancestry live around the world. The majority of them are in the United States, in North America, and a significant number in Latin America, particularly in Brazil, and in Europe, in France and in the UK. And those people, they are a very diverse group of people. They speak multiple languages. Inside Africa, there are more than 2,000 ethno-linguistic groups. They do have different religions and cultures, including Christianity, Islam, and traditional local religions. I will not go into a kind of huge discussion about what constitutes the African genome for the modern African populations. But we heard from this morning that people actually started in Africa, or the humanity started in Africa about 500,000 years ago. And through a lot of mixing within Africa, and then 100,000 years ago, some small group of those people migrated from Africa and populated everywhere around the world. And since then, the migrations back and forth from Africa continued to shape the modern day populations who are related or from an African ancestries. I just would draw your attention to even the near historical times, which is the last 5,000 years, where slavery, this is the slave trades, from the Arab slave trades, the trans-Saharan slave trades, and the transatlantic slave trades shaped the people who are from an African populations around the world. In addition, there was a lot of genetic flow from South Asia and genetic flow from the Europeans who populated Africa as well. And that will say something for the modern structure of the modern populations from an African ancestry. So it's mixed, it's diverse, and it's very, very rich structure. So we've seen this slide several times this morning, so I just wanted to reiterate the out of Africa theory is quite strong because it will tell us that a significant number of people around the world will have a trace, a little bit of an African genome on them. And then this slide again, it just summarizes what we've discussed this morning about the diversity of the African genome. So the African, and I would like just to concentrate on 3 main points. So the African genome is rich because it has unique variants. And these unique variants, they're large in numbers. So we're talking about quantity and we're talking about quality. And when we look at the populations from an African ancestries, they are diverse within themselves, so they are different. So as we heard this morning, there is a huge diversity between the east coast of Africa and the west coast of Africa. In addition, there is a unique characteristic, which is the LD pattern within the African populations. And this LD pattern is very important to study complex diseases like the neurodegenerative conditions, because it's a tool by itself. So using the LD blocks and the haplotypes block, which are shorter in the African population, I consider them as a very strong genetic tool. When you combine the genetic or the African genome or cohorts from an African background with other population, you can leverage this information to identify causal variants. And we will see examples from this in the work which resulted from our consortium. And we've seen this slide again this morning. The African genome is not just diverse. The African genome is not really rich in structure. The African genome is even bigger than the average reference genome. They're about 10% more or 10% larger than the reference human genome. And that chunk of DNA, which is larger and bigger, it has function and elements to it. It has about 315 distinct proteins. Till today, we don't know what is the function of these proteins. And we need to do more work to understand the relevance of this finding from the paper which has been published by Sherman et al. In 2019. Okay, so taking all this into account, let us see what do we have here. There are disparities in genetic research. This is a known piece of information. But what we know that is this will result in health disparities, which we could avoid. And we need to address this urgently. If we look at the GWAS diversity monitor, which is registering the number of people from different ethnicities who are contributing to the genome-wide association studies, as for today, the African people from an African background represent 0.18%. So we cannot allow that 18% of the world population to be presented by 0.18%. And this is in comparison to 95% from the European populations. And by far, they are the least represented groups within any genetic studies around the world. So this health disparity obviously is very obvious, is quite wrong for these reasons, for scientific reasons, for economic reasons, because if we do not know how to address the health issues of 18 to 25% of the world population, this will come with economic burden. And for the moral reasons that we discuss, is not just and is not addressing equality and equity. So that's what brings us to the Parkinson's disease. So Parkinson's disease, I will go quickly through the symptomatology and the genetic background of Parkinson's disease. Is a progressive neurodegenerative condition. Unfortunately, there is no cure, and the treatment is only symptomatic. Is the hallmark of Parkinson's disease is what we call Lewy body, which is a very proteinaceous material. It's like inclusion body, which is infecting basically the neurons and cause neuronal terminal death, and its effect basically is a substantia nigra in the brain, but it can involve different parts of the brain and result in manifestation which we can see in patients. These are the motor symptoms, including tremor, bradykinesia, falls, and rigidity, but it has other mask symptoms which we can't see in patients. We call them non-motor symptoms. These include cognitive decline, sleeping disorders, mood disturbances, and anxiety and pain, and is a disease which is felt by the patient because it's progressive, but felt by the families and the communities around the patient. And the burden of the disease is on the rise, is increasing for several reasons, including we're living longer, the number of people who are populating the planet is larger, and we're good at diagnosing the disease as well. So we know that the number of people who will be affected by Parkinson's disease will double by the year 2040, and it's not a disease of Caucasian and Europeans only. It can affect all ethnic groups, all around the world, and it can affect young and old people. So when we look at the African continent, for reasons we know, that the continent, the populations of the continent in Africa is increasing, and the life expectancy is increasing. So it's increasing by average of about 15 years in the next 50 years or so. And we know as a result of this, the burden of neurodegenerative condition will increase. This is include dementias and Parkinson's disease. The global burden of disease study estimated that the prevalence percentage change in age standardized ratios in Africa, this is the rising figures in Parkinson's disease despite of the age. It will be anything between 10 and 40%. 10 and 40% is double the figure of what we're expecting for the number of patients in Europe and America. So obviously, that in the next 50 years, the bulk of patients from Parkinson's disease, with Parkinson's disease and other neurodegenerative condition will not come for the Northern Hemisphere, will come from low and middle income countries, particularly in places like Africa. This slide has a very, very small font, so I apologize for this, but I tried to capture 200 years of history for Parkinson's disease in one slide. So Parkinson's disease itself has been described by James Parker in the 1917. And if we look at the last 200 years, we can see the progress within the last 30 years is even better, and it's faster than what happened in the beginning 170 years. So some big landmarks that we need to draw the attention to the audience too. So in the first 30, 40 years, as the Lewy body disease by Frederick Lewy has been described as the hallmark pathological mark of Parkinson's disease. In the mid of the last centuries, in the 1960s, L-DOPA was described as the first effective treatment for Parkinson's disease, and not a lot of progress happened until about 1997. So in 1997, the alpha-synuclein mutation was described, and this was a landmark in the Parkinson's disease field, because it's identified the whole pathological mark of Lewy body disease, which is the alpha-synuclein, which is a protein which is misfolding and build inside, and it results in building of these proteinaceous materials inside the cell, and it's linked to a genetic mutation. So we knew that there is a link between the genetics and Parkinson's disease, and since then, multiple genetic studies identified multiple genetic causes for Parkinson's disease, using for both monogenic, this is familial types of Parkinson's disease, and non-monogenic. So these are things which are identified. These are mutations and variants which has been identified through genome-wide association studies. And this opened the field to huge and advanced developments. So now is a very exciting time in Parkinson's disease science and studies. So is expanding. So we started expanding to GWASs, functional studies, building database, thinking about biomarkers, and designing therapeutic trials, which is focusing even in the disease management, not even symptomatic management. It's disease modification rather than a symptomatic management. So this slide summarizes the number of genes which we know that is associated with Parkinson's disease. So if you see in the boxes, in the black boxes and the blue boxes, these are genes which are associated with the Mendelian types of Parkinson's disease, and they run in families, and they're always associated with younger age of onset and severe progression. And I will mention some names of those genes because in the next slides I will be mentioning these names quite frequently. One of the famous genes which has been identified early is the MAPT and the GBA and the LRRK2. So if you could remember those names, we will bring them and we'll discuss them in details in the next slides. And then the number of those genes is about, the numbers which are associated with Mendelian inheritance is about 23 genes or so. But these could identify the genetic risk in about 5 to 10% of people with Parkinson's disease. So how we will be able to understand the heritability of Parkinson's disease and how we can improve on this. So this is when the GWAS study came. So GWAS looked at genetic variants across the genomes. These are millions of genomes, and looked at common variants. These are things which are common in more than 5% of the populations, and we looked at if these variants in combination can increase the risk of individuals to have the disease. And this study indeed has been really very, very successful because it helped us to understand or improve our understanding for the heritability, and we've been able to resolve up to about 30% of cases. So we understand the heritability of Parkinson's disease up to 30% now based on these GWASs. In addition, the finding from GWASs help us to understand things related to the disease progression, help us to stratify subtypes of the disease, help us to understand the molecular pathways using techniques such as enrichment analysis for pathways. So we know now that certain pathways, such as lysosomal pathways, mitochondrial pathway, endosomal pathways, and immune responses, are definitely associated with Parkinson's disease. In addition, we've been able to use these GWAS signal to understand the gene-environment interactions and what sort of environmental risk factors can increase the tendency of someone or a population to develop the disease. So they have been really, really very successful in helping us understand a lot about the molecular background of Parkinson's disease. And now the finding from these monogenic studies and the GWASs, we started thinking about translating them into clinical trials. So certainly for two genes, the GBA and the LRRK2. So they are in different stages of clinical trials in different phases, phase 1 and phase 2, and some of these clinical trials are in phase 3 now. So we're now in the era where we're heading from symptomatic therapies into disease-modifying therapies using finding from genetic studies. And these are the largest GWASs, which has been published in Parkinson's disease up to date. The largest is the one by North et al., which was published in 2019. This was done in the Europeans, and it's used about 37,000 patients and nearly 1.7 million controls. And it identified about 79 loci as risk, as associated with increasing risk for Parkinson's disease in the Europeans. There is another GWAS, which has a smaller GWAS in about 8,000 patients, and I think it's about 10,000 controls in Asians, and replicated some of the finding in the Europeans and identified 2 new loci in Asians. And a smaller GWAS in about 1,000 patients or so in Latinos, and again replicated the most solid finding in Parkinson's disease genetics, which is the association between alpha-synucleins and Parkinson's disease. And more recently, there is a combined meta-analysis of trans-ethnic GWAS using Latinos, Europeans, and Asians. But till recently, there was no GWAS in Africans. In fact, this work has been done by our consortiums. When you look at the Parkinson's disease research across the continent, you can see that the research is generally poor. In the last 100 years or so, the number of studies in Parkinson's disease, whether clinicals or genetics or epidemiological, they are very, very small in number. We managed to identify only about 180 studies. Half of them are genetic studies, but those genetic studies has been done ad hoc as in replications with no new finding which identified any mutations related to patients from within the continent. And this work has been actually replicated on another review article which has been done in collaboration between our group and a larger group of Parkinson's researchers within the global Parkinson genetic program. So we look at all the genetic studies which has been done in underrepresented groups, and by underrepresented groups I mean people who are not Europeans or Caucasians. And so the underrepresented group will include people from Asia and Latin America and people from an African ancestries. And by far, the lowest numbers of genetic studies were done in people from an African ancestries, even within the underrepresented group. So only 4% focus on the genetic research among people from an African ancestries. And this is focused on the north part of the continent, in places like Morocco, Algeria, and Tunisia. And the reason for this, because one of the genetic mutations which has been identified in Europeans, the G2019S, has a higher frequency in North Africa. And as a result of this, a lot of research was done on that population. But beyond the G2019S within the LRRK2, we didn't know anything about the Parkinson's disease, especially in sub-Saharan Africa. So this is where our consortium came to play. As I said, we launched the consortium in 2019. Our mission to improve this dire situation by conducting clinical and genetic research and engaging with the community to increase education and training and capacity building. We were funded by the Michael J. Fox Foundation, and we have a strong partner with an international consortium called the Global Parkinson Genetic Program, and we received some funding from the UCL Grand Challenges. As for today, we are about 140 collaborators in 55 institutions among 16 African countries. I don't have time to tell you about the details of the consortium, but we publish a correspondence which has been published in the Lancet Neurology in 2021, so I encourage the audience just to read about the details of the consortium. As I said, we do have a collaboration with the Global Parkinson Genetic Program. It's a very ambitious international program aimed to genotype about 150 individuals from across the globe, a significant number of them, about 50,000, from diverse cohorts from around the world. As for today, our consortium managed to recruit, so our target is 8,000 patients. We managed to recruit 8,000 patients on control. We managed to achieve 75% of our recruitment target, and we just started analyzing our cohorts. So the largest cohort that we have to date is coming from Nigeria and Egypt, and I will talk to you about some of the preliminary results from Egypt, and then I will discuss the major finding from our consortium, which is basically coming from Nigeria. So this is the preliminary results coming from Egypt. So we looked at a small cohort from Egypt of about 500 patients, 500 controls. We looked at some of the genetic variants which has been identified in the Europeans, including 13 genetic variants in the LRRK2 gene, which is the strongest gene, which has been identified several times in Europeans. We looked at the MAP gene, which has been identified, or the haplotypes within the MAP gene, which has been linked to the Europeans, and we looked at the ABOE as well. And understandably, because Egypt is a mid-between the Middle East and North Africa, and within the migration route between the Middle East and North Africa, especially within Israel, the G2019S has been identified in that cohort. And so the highest prevalence of the G2019S was found in people from an Ashkenazi Jewish background and in people in North Africa. And so the percentage of people who have Parkinson's disease, or the prevalence of G2019S in Parkinson's disease in Egypt, is about 5%. This is quite high because the prevalence in the European is anything which is 1% and low, and lower than that. Is not as high as North Africa, understandably, is not as high as the Ashkenazi Jewish groups in around the Middle East region, but probably it's linked to a migration from the Middle East all the way to North Africa. So we definitely saw a trend of a higher prevalence of the G2019 there. We replicated the map in our cohort as well, and it's a gene which is associated with Parkinson's disease in the Europeans. So we could see that there is an overlap between the genetic architecture in Egypt and the genetic architecture in other populations, including the Europeans. When we tried to do the same work in Nigeria, we couldn't find any of these genetic mutations. So in sub-Saharan Africa, there are no genetic variants in the LARC2, which is linked to Parkinson's disease. MAPT is not associated with Parkinson's disease in Nigeria. So we knew definitely the genetic architecture in West Africa is significantly different from the North African and is significantly different from the Europeans. And this brings us to the main finding from our consortium to date. This is a very fresh finding. So the paper was published in The Lancet about 6 weeks ago, and is a significant amount of work because, and as you see, is a long list of researchers. So we've included 78 authors, 46 of them from African institutions. It's a huge collaboration between our team, between a team of Nigerian neurologists within the Parkinson Network in Nigeria from different sites in America, including African Americans, and 23andMe collaborated with us and including cohorts of African admixed. So in total, we've included about 1,500 patients and nearly 200,000 controls. About 2/3 of the cohort are from a Nigerian origin. And these are the people who work hard, dedicatedly for about 6 months to analyze the data. And behind this team, about 70 odd neurologists who recruited, who worked hard and recruited all the patients on the ground. So I've included this workflow. It's kind of a traditional workflow for a GWAS study, so I will not go into details about it, but I want to draw your attention to one fact in the middle. So when you run a GWAS, normally you analyze a cohort which you recruit from a center. And when you do the meta-analysis, you meta-analyze different centers together. In our case, we didn't define the cohort from the origin or the site of recruitment. We defined the cohort by the ancestor group. So we looked at all of our patients in one pot and we divide them into an African or African admixed. The African are those people are more than 90% African in relation to the African within the 1,000 Genome Project. The African admixed are those people are anywhere between 70% and 90%. And those African admixed normally kind of segregated in a PCA plot with people from the southwest of America or the Caribbean. So in total, we included 1,000 cases and 1,200 controls who were defined as an African in the first GWAS, and the African admixed are a smaller cohort of about 500 cases, is about 473 actually, and it's about nearly 200,000 controls from 23andMe. So we've done the first GWAS in African, the second GWAS in the African admixed, and then we meta-analyzed both. So the first GWAS identify a hit in chromosome 1. The second GWAS in the African admixed did not identify any GWAS hit, but when we combined both, we managed to intensify the signal in chromosome 1. And then after that, we've done a follow-up analysis with admixture and haplotypes analysis. We try to identify any nearby structural variants using short and long reads, and we've done some functional work, including enzymatic assays and expression trait loci. So this is the finding from our GWAS study. So it's a hit in chromosome 1 in a gene called GBA1. It encodes for an enzyme called glucocerebridase enzyme. GBA is associated and linked to Parkinson's disease in all populations. There are about 300 variants in the GBA, which is linked to Parkinson's disease. We know that these variants in GBA reduce the enzymatic activity, the GCase enzymatic activity, and this would result to lysosomal malfunction and possibly result in developing Parkinson's disease. The variants which has been identified in the Nigerian is not detected in any population outside Africa. So it's a similar scenario for what we've seen from the studies from the presentation this morning. So it's a variant with an odd ratio of about 1.5. An odd ratio in a GWAS of 1.5 is quite a high odd ratio. The variance itself is very common in people with Parkinson's disease from Nigeria, and it can cause the disease in both homozygous and heterozygous status. So it has an additive effect. And the number of people, there's about 13% of the patients who have been included in this study are homozygous, and about 40% are heterozygous. And it's increased the risk anywhere between 300 times to about 120 times. So in people, it depends on the heterozygosity or homozygosity status. Most interestingly, this table, as I said, is absent from any other populations. So you can't find it in East Asian, South Asian, and Europeans. And if you look in African themselves, the frequency of the variants increased significantly from the East Coast to the West Coast. So the highest frequency is in the Isan, in Nigeria, and the Yoruba. One fact I didn't mention in the previous slide is a non-coding variant. And we know that all the variants which has been identified in GBA before in Parkinson's disease has been coding variants. In a similar way, the haplotype of that variant increased significantly, is absent outside Africa, and increased significantly as we move from the East Coast to the West Coast of Africa. So we know that a significant number of people who have in our cohort have this variance either in homozygous or heterozygous status, as we said. We know that it can increase the risk of Parkinson's disease by anything between 300 to 120 times, but is also associated with reduced age of onset. So people who are carriers for this variance, they experience symptoms in about 3 years younger or earlier than people who are not carriers. And when we look at this variance in our internal database and external database, we found that the variance is positively, or the percentage of people who are carrying is positively correlated with the percentage of their African ancestry. All these factors together suggest that's most likely this variance has an African founder effect. And in terms of the magnitude of the effect of the variance itself is as strong as the strongest variance which has been identified in the Europeans. So at that stage, which is the log 2. So the top part of the diagram is the GWAS in the African and the African admix, and the bottom half is the GWAS with a similar number of patients, reasonably similar number of patients in the European. And you can see the signal which is Rizig genome-wise significant at that level is the log 2, and we know the log 2 is the strongest signals in the European. So we knew that we found quite a strong finding. This is kind of a complicated diagram, but basically it's comparing the GWAS signals, the 90 GWAS signals from the largest GWAS in Europeans towards the signals which we detected in the African and the African admixed populations in our cohort. And as you can see, the structure of Parkinson's disease itself is different between different populations. But the most reassuring part of this comparison is the alpha-synuclein neurofinuculin, which is the pathological landmark of Parkinson's disease across different populations, is still showing the same direction and the same effect across all populations. So we knew that the structure of Parkinson's disease genetics across different populations, including the African and the African admixed and the European, possibly is sharing certain molecular mechanism, but at the same time is these molecular mechanisms are affected by different variants, but inherently it remained the same. These are the final few slides. So as I said, the variance itself has been identified in the non-coding regions of the GBA gene. So we went to do a whole genome sequencing and long-read sequencing to identify any structural variants nearby, and we couldn't identify any structural variants nearby. And then we've done some fine mapping to identify if the signal indeed is coming from this variant, and it came as strong as the causal variance in this locus with a serial probability of more than 70%. Then we expanded our work further to try and understand the functional relevance of this variance, and we look at expression trait loci and protein trait loci, quantitative trait loci, and indeed we found that variant is linked to increase the GBA expression. And this is a very, very significant finding, because if the GBA is affected, so in all the GBA variants which we looked at in the Europeans, the variants are linked to reduce the expression of GBA. In our study, the non-coding variants in GBA resulted in increase the GBA expression. And this was, at this moment, we felt confused. So we went further and we studied the enzymatic activity of the GKase enzyme in lymphoblast cells from patients. And as you can see, although the sample size is quite small, the GKase activity is reduced. So we ended in a place where we know that the genetic variance is definitely associated with Parkinson. It's causing increase of the expression, but is reducing the effect of the enzyme. And we hypothesized in the paper, most likely, the expression profile resulted in a faulty RNA. And that faulty RNA most likely does not get translated to the right protein. And this might result in reduction of the GKase activity. And this is where we are working now to understand more about the molecular mechanisms by which that variant is causing the reduction in GKase activity. So in summary, we find a new genetic finding in sub-Saharan Africa, which is linked GBA to Parkinson's disease in these populations. It the first variants which shows significance in Parkinson's disease in African ancestries. This variant is very rare in other populations and is a non-coding variant. Most likely it has founder effects in West Africa. And one important finding is it doesn't cause Gaucher disease, because GBA has been associated with Gaucher disease in other populations. But the prevalence of Gaucher disease in Nigeria is very, very low. So the number of patients that we have in our cohort is about 30%. So if Gaucher disease, if this variant would have been causing Gaucher disease, we would have seen more cases from Nigeria. So we're pretty confident that is not causing Gaucher disease. And it seems that it's exerted effect through a new mechanism, which we didn't expect and we didn't know about, and most likely through modification of gene expressions. And this possibly will open the avenues for new type of therapies in Parkinson's disease. So these are my last two slides. So this is the GBA function inside the cell. As I said, it affects the lysosomal functions. And in summary, there are a lot of therapeutic trials within to modify the GBA. function inside the cell. And this is by increasing the GKA's activities. And the treatments are focusing either in augmentation of the GKA's activity by a gene therapy, this is the GBA gene therapy, or restoration of normal intracellular function, GKA's function, through releasing the GKA's from the endoplasmic reticulum, or reducing the substrates, which is the lipid substrates which get accumulated inside the cell. If our finding is genuinely true, there would be a new molecular mechanism by which we can adjust the GKA's activity inside the cell, and this is RNA therapy, basically using something like antisense oligonucleotides-based drugs. And this is obviously a new mechanism. So our future direction is we will carry on doing more GWASs in other populations around Africa. Hopefully, we will be able to identify new variants which are unique to the African populations. We will work to identify the clinical phenotypes which are associated with these variants in African populations. We'll focus on things like cognitive declines and motor functions. We will continue doing some functional work to understand the molecular mechanism behind these variants, and we will continue doing more work, including imaging and biomarkers. And I hope at the end I convince you that studying the genetics of diverse population is not only important for moral reasons. Diversity in biology is quite important for scientific advances. And by studying variants like this, we will be able to understand more molecular mechanisms we would have never understood or found if we didn't include these populations. And at last, I would like just to thank my collaborators. I do have about 140 collaborators in Africa. My collaborators outside Africa, which is even larger. You can see that large number of institutions contributed to these studies. And I want to thank my funders as well, the Michael J. Fox and the GB2. And last not least, the patients who devoted their time, their efforts, their samples, and contributed to this work. And thank you very much.